Hello and welcome. This is Miss Fenton from Stevenson High School in Lincolnshire, Illinois, and welcome back. I'm going to start off today a little bit like we did yesterday with a quick overview of the exam for 2020. We have some news today, and we got it last Friday. More news is coming, more supports, more explanations. I'll look forward to that in later April. But the biggest news, of course, is mark your calendars. Tuesday, May 19th, is the 2020 AP Psychology exam. It's going to be online. And all of the tests are going to be simultaneous. So my students, we live in Central Time. So we're in the Chicago area. We're going to be taking our test at 1 p.m. If you're in Hawaii, you're going to be taking the same test at the same time, but it'll be 8 a.m. Hawaii time. The actual FRQs are not that different than what you've been practicing all year, right? So the big difference, of course, is there's no multiple choice questions. But this year, we're going to have 2 FRQs. And again, there's going to be a simulation and a video and some instructions coming out in April, later in April. So question 1 is a concept application question, and you're going to have 25 minutes to answer that question. And that question, when it's done, that 25-minute time period is up. You'll have 5 minutes to upload. And after the exam is uploaded, question 2 will begin, and you'll have 15 minutes. And that's a research methods question. These are both similar in structure to the traditional ones. So these are the ones you've been practicing, they're the ones that you've been having on your unit tests. You've been doing these all year. So I'm going to show you like a sample of what the College Board means by concept application and what they mean by research methods question. So a concept application question would look something like this question, question 1 from 2016. Now, concept application questions are normally 25 minutes, so the time limit isn't changed for this year, but I don't have confirmation if there's still going to be 7 points or not. There could be fewer, but I doubt that there would be more. But again, I don't know at this point, so that hasn't been released. But the time limit will be 25 minutes, so you want to start practicing taking a question that is of this length and answering it in 25 minutes so that you won't have that stress doing it for the first time under time conditions. What's great about this sample question, if you'd like to do it, the rubric, you can find it online on College Board, is that all of these points on this question come from units 1 through 7. And remember, this year, we're only going to have content on the exam from units 1 through 7. So this question, there is nothing here that's related to units 8 or 9. A research methods question might look like something like this. So they're going to give you an example of a study. And they're going to ask you some questions related to the data that's presented, asking you to draw a conclusion about the study. If it were an experiment, which this is not, they might ask you to identify the dependent variable. They might ask you about random assignments. You could be asked about statistical significance. So you're going to have to interpret this data. And oftentimes the research question has a part A, a B, and a C. And sometimes those later parts are a little bit closer to a concept application question. So in 2018, this question had a part B and a part C that were both more concept application related, although it was within the context of the earlier survey. Now, these first 2 terms in Part B, de-individuation and normative social influence, those are from Unit 9. So if those don't look familiar to you, don't worry. We're going to go over that unit later. Your teachers are going to make sure that you have exposure to social psychology as well. Now, some broad ideas about what it means to have an open note test and how scoring is going to work. There's a lot of questions going around. I know my students have questions too. The scoring is still going to be completed by AP readers, which are college psychology faculty, and AP readers can also be experienced AP psychology teachers as well. Every student will be given a score between 1 and 5 on their exam, and it will be completely based on that free response question. And what's going to happen is the student responses from the exam are going to be provided to teachers by May 26th. So your teacher or your school administration may choose to use this as some sort of a summative or a final, or maybe they'll just use it as formative feedback to help you improve. Also, I want you to know that the College Board has announced that they're going to try to release your scores as close to that typical early July date as possible. So you should look for your scores in early July. Okay. This is open note, open book, and you need to be aware that just because something's open notes, it doesn't mean that it's easy. Success is more than just copying information from your notes. Looking up facts is not enough. When you think about sodas, the apply in sodas, that's the most important part, right? So you're scored on your application. That definition is there to help maybe guide you, make sure that your application includes all the points, aspects of a particular concept that you need. Sodas, by the way, is not at all required. It's optional. That's just one way. If your teacher showed you a different way to organize your essays, that's fine as well. You can handwrite or type your responses, and either way, you'll have to upload that question in that 5-minute upload period. Once you start question 2, once you go to that research question, you won't be able to go back to question 1. Once you've uploaded it, and it has to be uploaded before question 2 begins, you will not be able to go back and forth between the questions. Okay, I need to be clear here. You still need to prepare for this exam. I don't want you to think because it's open note that it's easier. You can have resources and sure that may save you some time in memorizing, but you need to know how to apply the information efficiently and correctly, and that's going to take preparation. If you don't know what these concepts mean and you're having to look them up, that's going to really cut into your time. And if you're talking about a 15-minute time frame for that research question, you actually have to know what those words mean when you go in. So I want you to prepare for this exam just like you always would prepare for an AP exam. The fact that it's online should not make a difference. You also have to factor in the time it's going to take you to read the prompt, right? To read and organize your thoughts around that prompt. So you're going to have to answer complex questions in a limited time. And the more time you have to spend looking up information, the less time you'll have to work on writing your response. And remember that response, that application is what you're going to be scored on. The AP exams this year, they've been designed knowing that you have access to your notes. And so questions are going to ask you to apply concepts in new ways, right? So again, if you don't know your material, that's going to really, really slow you down. Okay, so collaborating with others is not acceptable during the exam. And the internet, while it's a great source for facts, it's also full of misinformation and diverging opinions and manipulated content. And basically, College Board has found that in the experience of university faculty, students taking open note exams who try to find the answer on the internet often end up earning fewer points than students who use their own notes, right, and have prepared with their learning. We strongly advise, the College Board strongly advises students to organize your materials prior to the AP exam so you don't waste time searching for information. And again, you're going to want to prepare. Exam security is very serious, and the College Board has many security protocols in place. What resources are allowed? Your class notes, resources from your teacher, previous assignments that your teacher may return to you. During the exam, however, you can't communicate with other people through any means, online, in person, texting, chatting, any possible way. All of these things are not allowed for the exam. If you have more questions about the exam structure, you can go ahead and go to this website. On the left side, you can click on a box that takes you to the schedule. And the section on the other side where it says taking the exams, that's going to give you a lot of details. A lot of things I just covered, a lot of frequently asked questions are going to be answered for you right there. And this is the place that you're going to want to go for upcoming updates. Okay, it's time to get started and talk about the FRQ from yesterday and look into the effect of drugs on neurotransmission and technologies for studying the brain. So yesterday, I gave you this study, this practice FRQ, and it has 3 parts to it. And it's mostly research-based, but it has a little bit of concept application at the bottom. I guess there's 1 research question and 3 concept application. Well, actually, part B is also research. So I want to emphasize for you here that applications are what score on the AP exam. And definitions can only be considered if they support an application. So to ensure that you score a point, you want to focus more on that application than the definition. The idea of writing a definition first in SOTAs is that if a term is really complex, maybe you want to sketch out ahead of time a definition so that your application includes all of the moving pieces of that complex concept. So part A was asking you, what is the most appropriate conclusion that can be drawn from this study? And the first thing you want to think about is what kind of study this is. And what this is a survey. And surveys are descriptive research. So because this method is a descriptive study, it's going to describe, and you want a synonym for descriptive describes, you want to record what's happening, right? So in a survey, descriptive research, you're gathering up information, you're naming or illustrating what's being observed. This isn't an experiment that would lead to cause and effect. So if your response said cause and effect conclusions, even if you use language like if then, leads to, makes, due to, or results from, right? Any of those types of phrases is implying a causal conclusion, and you cannot do that because there is no random assignment or control group here. And actually an experiment in this particular area would be difficult ethically because you would have to randomly assign minors to use cigarettes, right? To try e-cigarettes. So part B, we have dopamine and the reward center. And dopamine, the neurotransmitter, it has a lot of functions, right? So it's related to pleasure, reward, movement, learning, attention. And they're definitely present in the areas of the brain that produce pleasure, right? Wanting. The application, if you need to create an experiment, because it is an effect, right? The study of the effect is in the directions, then your application has to reference an experiment. And when you have an experiment, you want to include that random assignment and a control group. So researchers could create an experiment in which participants are randomly assigned to inhale nicotine or placebo, there's your control. And then they can measure the amount and location of dopamine activity in the brain and ask participants to rate their feelings, right? Like how pleasurable was the experience. And it is also important to note in your application, the order, right? The person has to intake the nicotine. That's going to then release dopamine. And then finally, a feeling of pleasure. You don't want to say reward because reward is in the term that you're being asked to define. So it's really easy when you're writing under time pressure to write, dopamine. The participants inhale nicotine that causes the release of dopamine, and then they feel the reward, right? So if you catch yourself doing that, you just add pleasure after the word reward. Just make sure that you're covered. Okay, part C. Humans display various reactions that are characterized by opposing tendencies. So this is what you could potentially see. It's a more sophisticated, nuanced example. And what I did here is I combined the definition and application to see how you could see how that would look, right? I'm giving you pretty detailed definitions in these videos because I'm also trying to reinforce content knowledge as well. So the opposing processes in a neural firing are potentially depolarization and repolarization, right? During the action potential, you have the ion exchange that leads to depolarization that sends the message down the axon. This is what we talked about yesterday. That's the action potential, but then the cell must repolarize, the refractory period. That's when those sodium-potassium pumps restore the ion balance and the cell is ready to fire again. It returns to that resting potential. In terms of neural firing and opposing processes, you could have gone a different direction. You could have talked about excitatory versus inhibitory postsynaptic potentials. You could have talked about excitatory versus inhibitory neurotransmitters as opposing processes. So you could have even gone with the action potential, the release of neurotransmitters into the synapse, and then the opposing process of reuptake, right, or breakdown in the synapse. So this gives you, this neural firing in this part C, a variety of ways that this can be answered. Also in part C, we have the autonomic nervous system, and here you have to have the two pieces. So you're going to have the peripheral nervous system, and that the two parts of it that are opposing are sympathetic and parasympathetic. So in your application, the autonomic nervous system's two divisions have opposing functions, right? So what you want to do, however, because this is a nervous system, you want some sort of physical response, something physiological. So if you just said arousal or nervousness or stress or potentially even fight or flight, it might not be strong enough to score because you're not indicating that physiological reaction. And the same thing with parasympathetic. So rest and digest is a mnemonic to help you remember it, but this is a nervous system function, and you should include something like a decreased heart rate, like for arousing. So again, with sodas, no intros or conclusions. This isn't a thesis. It's not a essay. If you can, space out your answers, like your different concepts, the different terms. This way, if you have to go back and add a little, you have some physical space to write that addition in. You want to answer in order so that you make sure that you're following the prompt. You want to ground yourself with the definition, right? So if you don't want to include that in your response, but I would still say, like, write out somewhere, think about your definition. What is meant by dopamine? What does dopamine do? And then apply. Reference that scenario that's provided or answer the specific question that's being asked. All right, let's take a look at the influence of drugs on neural firing. And I want to start out with a term called the blood-brain barrier. So basically, surrounding the brain and the spine is this semi-permeable network, right, of tightly packed cells. And it's designed to prevent poisons, right, from reaching the brain. And this was really interesting the way this was discovered. Researchers found that if they injected blue dye into the bloodstream of an animal, that the tissues of that animal's body would all be blue, except for the brain and spinal cord would not, right? So it was not crossing. Now, some drugs can reach the brain, right? Like anesthetics. Some drugs cannot, right? So some antibiotics can't cross the blood-brain barrier, which is certainly very dangerous, right? If there's a bacterial infection in the brain, you need to be able to get the medication into that, across this blood-brain barrier. Now we're going to talk about neurotransmitters and receptor sites. So this image here on the right, you can see at the top, you're going to see the terminal button of the presynaptic neuron, the sending neuron. And in there, you see those kind of like little bubbles with the little green circles. Those are synaptic vesicles, and the little green circles are representations of neurotransmitters, right? And that black area right there is really tiny. That's the synapse between the sending terminal button and the receptor sites, right, on the dendrite of the postsynaptic neuron. And those receptor sites, this is your metaphor, right? They're like the lock to the door of your house, right? And the neurotransmitter is the key. So if it's a dopamine receptor site, that dopamine neurotransmitter is going to fit in there exactly and turn that lock and open the door, right? Lead the action potentials begin. Now, there's two kinds of drugs, two broad categories. Agonist drugs work in a couple ways. And one way is that they're kind of like a counterfeit key. So it's not actually dopamine, right? It's a drug, and it's dopamine shaped enough that it works like a counterfeit key, and it's going to open that door. It's going to get that cell to fire. Antagonist drugs have the opposite effect. It's almost like a broken key that got stuck in the lock, right? And so the broken key isn't going to turn the door and open it, right? But it also is preventing the actual key, in this case, the neurotransmitter, maybe it's serotonin, from entering the receptor site. So let's take a look at the two ways agonist drugs work. So agonist drugs, this could also be a toxin, that they increase transmission, and they work in two ways. They either mimic the neurotransmitter, that's that counterfeit key I was just talking about. So if you look at the image where it says open, okay, you have the pink image that's opening it. That's a natural neurotransmitter. And then you could have an agonist, it looks like kind of like that green key. That's your counterfeit key that's also going to open it. And GABA agonists, there's a lot of kinds. Certainly they could be medications, anti-anxiety medications like benzodiazepines. It could even be pain medications like morphine, or even illicit drugs like heroin. Those mimic endorphins, so they would be those counterfeit keys. Alcohol is also, for example, a GABA agonist. And what they're going to do is they're going to cause the drug is going to cause that cell to fire as a counterfeit key. Agonists can also work in another place. It can also work in the reuptake process. So with antidepressant medications, they're called selective serotonin reuptake inhibitors, so SSRI. If you see in the close-up here, the SSRIs are the red kind of squares, right? And the serotonin is the yellow. So the neurotransmitter, the neuron's going to release serotonin into the synapse. And what Prozac, what that SSRI is going to do, is it's going to block the reuptake, which means that serotonin, the natural serotonin from the cell, is going to remain in the synapse longer, giving it a chance to then stimulate the next cell. So Agonist drugs like for Alzheimer's can also work in this way too. They are going to prevent reuptake and allow that acetylcholine to stay in the synapse longer. And they're going to maybe reduce memory systems. However, as we talked about yesterday, there is no cure. Antagonist drugs are that blocking, right? So they're like the key that got broke off in the lock, or maybe, you puts a metal piece in front of the lock. So the key physically can't get into the lock, right? So the neurotransmitter can't reach that receptor site because the drug is actually blocking it. And a really good example of this would be a dopamine antagonist in the form of an antipsychotic medication. So individuals with schizophrenia, they have too much dopamine. And so the medications that work on those positive schizophrenia symptoms block the transmission, right? By putting kind of something in that receptor site that prevents the natural dopamine from reaching it. And I mentioned earlier, these could also be toxins, right? So botulism, which is a serious type of food poisoning, what it does is it actually prevents the release of the neurotransmitter acetylcholine, and it can lead to paralysis or breathing problems, possibly even death. Okay, let's take a break here. We just covered a lot with our medications with agonists and antagonists, and I want you to take a look at the following 3 examples. Some of this we also talked about a little bit yesterday, so maybe you can remember a little bit more. Okay, so with example 1, you have schizophrenic symptoms that are associated with excess dopamine. So now you're going to need a medication to reduce it, right? So you're going to need a dopamine antagonist medication, which would be, an example would be an antipsychotic medication. With Parkinson's, these individuals have reduced levels of dopamine, so they need more dopamine, right? So now you need a dopamine agonist, and one of those is this drug called L-DOPA, which helps the body to make dopamine more effectively. Example 3, this is the one that we talked about in the previous example. So depression are often associated with low levels of serotonin. So you're going to need a medication, a drug that's going to increase that transmission. So you're going to need a serotonin agonist. Okay, so now let's take a look at kind of some historic and modern ways to examine the brain. So how do researchers study the brain? And we're going to start with a very famous historic case that maybe you remember from your biobasis unit. That's the case of Phineas Gage. So Phineas Gage suffered a horrible accident. He was working on building a railroad, and he had a tamping iron that went through his skull. And just to be clear on that image, the image is just frozen in time, right? So the tamping iron, which he's holding in the other image, literally went in one side of his head and shot out the other like a bullet. He was using this tamping iron to pack down gunpowder, and a random spark ignited it. So the surprising thing was that he was able to talk. He was conscious, right? They were able to get him to a doctor, but this is an era before antibiotics, and he survives. But what psychologists were able to do is examine the changes in Phineas Gage's behavior as a result of damage to his frontal lobe. So sometimes, especially early on, the way researchers studied the brain was by examining changes in behaviors and cognitions that occurred following an injury. 2 other individuals we're going to talk about. The first one is Paul Broca. He's one of the names in the curriculum that you're supposed to learn about. And he did an autopsy on a patient. So after the individual died, he went in and looked at his brain. So the patient, after suffering a stroke, was only able to say one word, right? So he basically just said tan. And Broca found damage to a large area of the left frontal lobe in the autopsy. And the association was made that potentially this area was responsible for the production of language, right? And this area is now named after Paul Broca. It's called Broca's area. And individuals who suffer from this type of condition, like what happened to this patient who could only say tan, is referred to as Broca's aphasia. So this is an area that's in the left frontal lobe. It's kind of near the motor cortex. And a person with Broca's aphasia, if this was damaged, right, would struggle to produce or speak words, but they would potentially be able to understand language. Another language area that was found in the similar way, and also through an autopsy, was found by this man named Carl Wernicke. And he found another area of the brain that's in the left temporal lobe that is related to language comprehension, right? So his patients could form words, but what they said did not make any sense. And the autopsy found the damage in this like left temporal lobe. Now, since Wernicke and Broca, research has demonstrated pretty convincingly that language involves several brain areas that are working together. It's more complex than this. Just to show you that we still study things today through autopsy. So there is, what's going on here? This condition called chronic traumatic encephalopathy. It's a degenerative brain disease, and unfortunately, the only way to accurately diagnose it at this point is through an autopsy after death, right? Now you can examine the effects of concussions, you can see that, but this particular condition, in order to study it and learn how to prevent it or treat it, the only way this can be done is, post-mortem through an autopsy. Now we also have imaging methods, right? So various ways to study the brain, and they kind of fall into two categories. So either they're going to examine structure, which is essentially like the anatomy of the brain, or the function, like the active working brain. And some of these, in the case of the fMRI, do both. Okay, so we're going to start with a CT scan or a CAT scan. And this is a type of imaging that is going to involve X-rays, right? So it involves radiation, which is a detriment because you can't, use this as a study tool really easily. You can't repeatedly give people exposure to radiation, but it is less expensive than the other scans, and it does allow researchers to see kind of large brain or abnormalities, damage from tumors. So it can be used both, diagnostically and somewhat as a study tool. The next one that's also a structure is something called an MRI. And an MRI of the brain involves computer enhancement, and it creates much clearer images, right, than you have with a CT scan. Plus, MRIs also have another advantage. They don't involve radiation, and they can view much smaller areas of the brain, and they can show more detail. So in the yellow here on this image, that's actually highlighted, that's the hypothalamus. And if you look, you see the red arrow pointing to a structure kind of behind the brainstem. I was wondering if you know what that is. So what that structure is it is the cerebellum. So here's an example of something like that they might use to study various conditions. So brain shrinkage, which is evidenced by enlarged ventricles. You can see that on the image on the left. And this is shown as a result of alcohol use disorder, right? So they're having this woman who has alcohol use disorder, and this is her brain, and she's being compared to a control condition, a woman of the same age without alcohol use disorder. So both the CT scan and the MRI, those are going to show researchers' structure, like literally images of the brain. Moving on to an example of function, you have the EEG. And the EEG is non-invasive. It doesn't involve exposure to X-rays. It's just these little electrodes kind of placed on the scalp. And it's often used in sleep research because it can detect kind of that electrical activity associated with the various stages of sleep. It can show abnormal patterns that might, identify neurological conditions. It has pretty good time resolution. It gets readings like kind of every few milliseconds. It is somewhat difficult to determine the specific brain areas involved, right? And it only has the ability to examine electrical activity kind of on the surface of the brain, the cortex. You can also use this method with something called an evoked potential. So these are amplified tracings that show changes in voltage when the participant who's part of a EEG study responds to a stimulus that's presented. So you can check these evoked potentials to see activity in different regions. Electrical stimulation is the advantage because this can show what areas, so like we call this idea of like a localization of function, right? So if you're going to do brain surgery, then it might be helpful for surgeons to know the areas that you should not cut, right? To not be near, right? So the potentially like language areas. Now, you can also use this to study animals, right? So you can stimulate the amygdala in a mouse and you will get aggressive reactions. So the amygdala is strongly connected to the emotions of fear and aggression. This can be somewhat invasive, and so the research is typically done on animals. And also this one doesn't show structure, this is just function. So another one that shows function that can use both as a method of study, but also interestingly, potentially as a treatment method as well for something like depression. So this is a non-invasive method that uses magnetic pulses and that can temporarily enhance or depress activity in a targeted region, right? So that temporary depression, like right, the preventing of the firing in that area creates like a virtual, like an imaginary lesion, right? And it allows researchers to create these virtual lesions and observe what happens in damaged areas without actually causing any damage. It is also being investigated as a tool for depression, schizophrenia, feeding and eating disorders, anxiety disorders. So this is what those kind of two processes might look like. So there's two different versions of transcranial magnetic stimulation in terms of depression treatment. Okay, so we already talked about the MRI. Now we're going to talk about an fMRI. So that small f in front of the MRI stands for functional. So this one's going to show structure, brain anatomy, using magnetic fields, the same way the MRI works. It's the same machine. But it's also going to track real-time brain activity by measuring oxygen levels and blood flow, right? So that's a metabolic process. It's going to give you real-time brain activity over seconds versus minutes. So it's a little bit faster than a PET scan, has better quality than a PET, which we're going to talk about it in a minute. There's no radiation involved. Disadvantages, it's the same as the MRI. This cannot be used on an individual with a metallic implant, like maybe a surgical pin. This is an example of some images from an fMRI. And this is what they're comparing here is the experience of pain versus imagined pain to try to study something like empathy in the brain. So brain activity related to actual pain, which you can see on the image on the left, is being compared with somebody who's observing someone they care about who's in pain, right? So the emotional areas related to pain show up in the empathy condition, but the somatosensory cortex, the actual physical experience of pain, is not being experienced in the empathy condition. So what's great about an fMRI for a research perspective is that researchers can communicate with the participant in the scan via an intercom. They could also visually project things so you can kind of see what it looks like when people are thinking about someone they care about or thinking about something that's stressful or they see an image that's surprising. And next we have the PET scan, and this one is a brain imaging method that involves kind of this injection of this harmless, short-lived, so it's not going to last very long, radioactively tagged glucose into the brain via the blood, right? And so the areas that are active during a particular task, those areas are going to burn glucose, and that glucose is essentially tagged, right, with this injection. And PET scans, they can be used for both the detection and the study of brain diseases. But these, because of that injection of the radioactively tagged glucose, they can't be used a lot of times in a row, right? For example, on a particular individual or participant. So some advantages is that you can see areas responsible for specific functions, right? So this allows that really detailed investigation into brain illnesses like Alzheimer's or epilepsy. And you can study even the activity of a specific neurotransmitter. And again, the disadvantages are that exposure to those low levels of radioactive material, there's a little bit of a lack of fine detail and poor time resolution. Okay, so now I'm going to ask you some questions about these scans, but I'm going to ask you these questions in the context of how the nervous system works. So if you're asked something like this on an FRQ, think about how you might answer it. So you're going to have to come up with the answer yourself and then explain why, right? So it's going to ask you to understand how the action potential works. And because that action potential is electrical, that allows us to study that with devices like the EEG or electrical stimulation. If you want to see something that's metabolic or biochemical in nature, then now you're looking at the PET, which is going to trace that metabolization of that tagged glucose, that radioactive glucose that was injected. And with an fMRI, it's about oxygenation in the bloodstream. Okay, so that's quite a bit for today. We reviewed the FRQ from yesterday, which had some research points and some concept application points. We talked about agonists versus antagonists and kind of some specific examples of how those might be used in treatment of various illnesses or diseases. And then we talked about some technologies and strategies that researchers use to study the brain and nervous system in conjunction with like how that nervous system works. So I'm going to leave you with a pretty lengthy AP exam practice. So it's a concept application question, but it has 6 points. And again, you want to focus on that application. You should do SOTAs. You should space it out. You should answer in order. You should think about including a short, simple definition. And then from that definition, you're going to make sure all the pieces of that definition are included in your application, because it's the application that the readers are going to be scoring. You want to make sure that you're reading the question carefully. So this is a pretty sophisticated question, and it's going to ask you to put together some things from yesterday when we talked about neurotransmitters like acetylcholine, and it's going to talk about some things from today. Myelin sheath is a part of the cell, asking you to kind of integrate these things, and you have that one research concept point up at the beginning. Also, for acetylcholine, it also says in parentheses ACH, so capital A, capital C, small h. Feel free to use that ACH acronym when you're writing, so you don't have to write out acetylcholine. One, it's hard to spell, and two, it takes a long time to write. And the same thing for positron emission tomography. If you want to call it PET or PET scan to save time, that's perfectly fine. So I really want you to take some time, try to even set a timer. Give yourself, 25 minutes to answer this question and see where that goes. If you go over the time, just keep going. But then we need to maybe talk about how we can shorten up the time frame that you need. Again, if you're having to look up these words first, right, that's going to eat into your time. So it's best that you prepare for this exam as if it were a regular exam. Forget that it's open note. You need to know these concepts. And one more time, this is if you're having any issues with technology or access to a device, or someone that you know who's going to be taking the test is having concerns, you can go ahead and give them this link to the survey and reach out to the College Board. Okay, thanks. Really appreciate you joining me today. I hope you have time to work on that practice question before tomorrow. We're going to definitely go over the answer and keep studying. Bye-bye.